Good morning, dear delegates and participants. A very warm welcome to all of you. Myself, Devashish Ghosh, Assistant Professor, Roland Institute of Pharmaceutical Sciences, Barampur, welcome you all from the bottom of my heart for the first scientific session of Day 2 EFDP on AI on drug development, recent trends and future perspective, sponsored by AICTE-ATAL Academy, New Delhi, and organized by RIPS Barampur. Before any auspicious start to good work, let's take the blessings of Lord Ganesha to make it a successful one by rendering a virtual prayer.
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
वक्रतुण्डमहाकाय सूर्यकोटिसमप्रभा निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा सर्वकार्येषु सर्वदा
Dear participants, now we have with us our most respected resource person of this session, Dr. Girinath G. Pillai, Scientific Officer, Zastra Innovations, Bengaluru. At the very outset, I would like to introduce Dr. Pillai to all of you. Dr. Pillai is currently designated as Scientific Officer, Zastra Innovations, Bengaluru. He is one of the coordinator for MHRD Drugs Discovery Hackathon 2020, founder of Zastra Innovations, Bengaluru, and visiting scientist at Nairo Research, India. He was a former Marie Curie Research Fellow at McLeod Limited, Estonia. He carried out his PhD at University of Florida with Professor Katriski and Professor Carlson, University of Tartu. He holds 40 research papers in peer-reviewed international journals, one US patent, and co-authored a book chapter. His expertise is in the area of machine learning, molecular modeling, softwares, pipeline development, KNIME workflows, mechanistic-driven drug discovery. Apart from the scientific positions, he is also a mentor of IIT Kottayam, Government of India, Niti Aayog, ATL schools, boss member and research advisor to few universities across the country. With this brief introduction, I would like to request Dr. Girinath G. Pillai sir to share his views and start his scientific talk on 2D and 3D lead optimization using computational tools. Welcome.
Thank you so much for the wonderful introduction, and thanking Dr. Biswa as well as the principal and organizers of this FTP program for giving me an opportunity. And this is my second time with this particular session, so I thought to do it a little bit different, still related to machine learning. So today we will be having first few minutes of presentation just to give an overview, and then I will get on to two different softwares on how to do optimization as well as how to apply machine learning in structure activity relationship studies. Whatever I'm showing you, there's a combination of both commercial tool as well as free tool. Both are available, so you have a choice to choose. And if you have any questions in between, please feel free to ask me. I know this is solely chat or a command, comment based interaction. So please post your comments so that I will be able to respond to you in between, uh, coordinating with the organizer. So, yeah, so basically, everybody was concerned about maybe 4 or 5 years back, mainly about docking, virtual screening, QSAR, pharmacophore, and all. But recently, we could see a lot of trend happening on molecular dynamics as well as optimization. We have learned a lot about lead optimization, but have we really applied in silico tools to do ideation or decision making on lead optimization? It's a big question mark. Most of the medicinal chemists and organic chemists, they try to do optimization based upon their expertise, and they're best in doing that. But sometimes these tools will be able to give you some more additional thought process or ideas which we might not have came across. As a human, we have a limitation on getting or memorizing a lot of information. But computers and databases, of course, if it is used properly, it has a more memory and retrieval power. That is exactly what we are trying to look at today. How these reaction databases can be explored, and also synthetic feasibility can be looked into the lead optimization. And this is very important nowadays because everybody is looking for novelty, even if it is publication or if it is patenting. Novelty is a big factor. Now, novelty is not only based upon process or protocol, but also on the product itself. Later, if the outcome of this may be an biological activity or chemical or physical property, whether this is going to be better than others or not, novelty is something that is everybody is looking at. So, I want to just start with a disclaimer that we are not yet completely ready with AI or ML in drug discovery. It takes time, like the time taken for Human Genome Project. I don't say that we have stories came up with the AI in drug discovery. We still have. But can I use it or can you use AI in drug discovery today? No, right? It's so expensive for us to take care of that. That's the reason why I said it's not completely ready. It is still in its initial stage. Many teams and many big groups are able to make use of it and see how far it goes. And the IT infrastructure is expensive for AI. But what I would say is ML in drug discovery is still there. Back in since 1990s, it is still there. But we did not give much importance to the word ML. Rather, we used statistics. Rather, we used empirical methods. But right now, there is a hype in these terminologies of AI or ML. That's why we even the basics of ML is statistics, but we don't talk about that. We just say ML, right? So today, what we are going to focus mostly on how machine learning techniques can be used in drug discovery when you're doing 2D or 3D based optimization to make decision which compound I have to synthesize, to make decision which compound can give better potency, to make decisions on which compound can have optimal pharmacokinetic properties and less toxicity. And this is something very important. My slides might contain pictures and videos taken from articles, lectures, and other tutorials. So the copyrights and the authority goes with the respective authors. The slides that I'm presenting today is available in the slideshare.net Giribio, and few other demos and others are already available within these my YouTube channel. And today's workflows, which I'm going to show in RapidMiner as well as in StarDrop, is available in GitHub. So RapidMiner is a freely available software. which you can use it for machine learning models. Only thing you need to have the data with you. It can train your data, it can evaluate your data, as well as deploy your models. These are possible, which I'll be showing you today. Now, again, this also I am repeating it again. I repeat this in every presentation. We always say bridging the gap between by connecting between industry and academia, but ultimately, just the connection is not enough. The academia has to understand what is the expectation from an industry, not in the perspective of placing a student, not in the perspective of employment. Yes, that is an important factor, but on the perspective of what is viable for commercialization. Now, wait and see. Many of the funding agencies, government agencies already started, but very soon everybody is going to implement this. If they wanted to fund you, two things will be required. Number one, what is the novelty that you're bringing in? It's not about publication, it's about technology licensing, which I mean to say is patenting. Patent means money, that's commercialization. So patent is not for putting into the CV, it's mainly for commercial. Can you make money out of it? So funding agencies are going to propose this. Can you commercialize whatever you're going to propose in this research? What is the outcome is? What is the technology viability? Can it reach the market? And this is the expectations from the basic research that can come from academia. And number two, what is that the what to say relevance of study? That's something very important, right? Academic, being it's a non-profit and it's for education and awarding degree, many of the things are being available to you free of cost, having better access than industry, and also more team work with the research perspective, right? So they look at academic research, how far you can do extensively so that the technology can be transferred from academia to industry. And that is where the industry takes care of commercialization. So this is the place where we have to look at the bridging the gap. Now, if we say, sir, I'm not so advanced yet, I'm not yet there, the question is when you will reach there. It has to be as soon as possible. There are many others are doing that. It's not that we also have to do the same way others have done, but we have to enrich or we have to flourish our findings to the next level of technology transfer. And that's where, when we talk about being this is a FDP again related to drug discovery, let us imagine how AI or ML can help a normal civilian in some way or the other. Number one, how to reduce the cost of the medicine. The main implementation of AI ML is to do cost effective research or cost effective decision making. So many of the machine learning models are helping you to make decision making. See, simply giving a data and generating a model doesn't make sense that, okay, this is going to be useful for 100 people. No, until unless you deploy it, until unless you make it public or make it others to use it. Okay, this model can be used and used for predicting the activity or property for their set of molecules. Until unless then the ML models are useless, right? Just for publication, if you're saying I published on ML, okay, how many people made use of this publication? So that's where we have to understand where to apply and deploy. And that will be discussed today during the demo session, which is very, very important. So ultimately, a civilian thinks that is this particular drug is going to save me from my illness? Second, is it going to be affordable for me? But in many cases, you can see most of the cost of the drug is very much high, and its high cost of medicine is sometimes not affordable for many of the civilians. So, what you can really do in order to reduce the cost of medicine? Let's analyze that. In order to reduce the cost of medicine, you have to reduce the cost of failures, not only on oral drugs, any others. But I have an example for oral drugs. Between 1960s and 1980s, we do have many FDA approved drugs as well as rejected drugs, especially oral drugs. So these two reviews actually looked into what is the main reason behind the failures of these oral drugs. Surprisingly, they were able to find that the pharmacokinetic ADME property was having a major share in the failure of drugs, about 40%. And later, efficacy, which is the phase 2 of the clinical trials, is also one of the reasons for the cause of failures. Surprisingly, later 90s or early 2000s, we were able to reduce this cause of failure by implementing structure activity relationship. Oh, sorry, I didn't replace my graph. Okay, that's my mistake. So on the later stage from between 2000 to 2010, we could see that the peak ADME and efficacy got reduced. This is a wrong graph pie chart. I will show you once I finish this slide. So the later you can see the strategy and toxicity has increased. The peak ADME and efficacy contribution to the failures has reduced dramatically. So, strategy, how can we minimize the errors and how can we reduce the toxicity? That's exactly what we are going to discuss today on 2D and 3D lead optimization. Now, today's agenda is what is AI and machine learning and what is the application or implementation of ML in chemistry and what are the implications of drug discovery on how to avoid the failures as well as what to do next or what to expect in AI ML. The first part, when you look at machine learning, learning denotes changes in a system that enables a system to do the same task more efficiently the next time. It's like a human, right? We try to make mistakes, we learn from the mistakes, and the next time when we apply, we make wonders. We try to implement it much better way. It's the same way what Herbert Simon mentioned. So when you're trying to apply these ML models, it tries to learn itself and it comes up with a better enrichment. And this is really going to help you how to improve your models and optimize better. And later looking at artificial intelligence, it's a combination of both machine learning and deep learning. And when you look at machine learning is not artificial intelligence. I'm not going too much technicality into that because I can understand most of the participants are from pharmacy or bio related or chemistry related expertise. So it's better not to go into too much of coding and too much of algorithms rather than we will understand how this can be applied somewhere. So, machine learning is a branch of computer science which deals with system programming in order to automatically learn and improve with experience. So, in statistics, that's our advantage here when compared to statistics. In statistics, of course, we are trying to model it, we are trying to correlate it, we try to understand about its descriptive data. But when coming to machine learning, when you're giving additional data, we are able to learn by, or the program is able to learn by itself, or the model is able to learn by itself. That is advantage here. So the roots are being programmed so that they can perform the task based on the data they gather from sensors, and it automatically learns the programs from the data. So this is how. So they try to get additional data and perform accordingly. But there's a big challenge recently came in for robots, which I will mention it a bit later in detail. I won't tell which is the company and all. They stopped that project of robotics just because two different robots started talking to each other because they are not made for that, right? So there is some alarming things also involved, which we have to look at it. Anyway, that we will discuss a bit later. Now, these are the different machine learning algorithms available and which are being applied at different segments or different level of analysis. Mainly, machine learning started looking at big data visualization, identifying fraud in the credit card or any other data, then learning task, how to optimize e-commerce, which product that you might like, or customizing the advertisements, all those things are being taken care. But what are the types of techniques in machine learning? Supervised, unsupervised, semi supervised, and reinforcement learning, including transduction and learning to learn. The popular algorithms in machine learning are decision tree, neural network, probabilistic networks, nearest neighbor, and support vector machine. So we will be looking at these few of the algorithms today, including decision tree, neural network, and support vector machine, including ADA boost. So these are the things that to be taken care, which usually these algorithms are going to help us in structure activity relationship when we are comparing between the molecular descriptors as well as the activity that we want to predict. Now, when you're looking at the stages of ML, not only the stages of ML, even in stages of statistics are also the same. You have to first build the model, validate or test the model, and apply the model. In many a times, we do not apply these models. For example, when you publish a QSR paper, you build the model, you test or validate the model, but do we apply somewhere? Or do we give an option for the reader so that they can you make use of your model so that they can predict the activity for their compounds? So we are not really applying the model, right? You might have applied by yourself in your paper, but how it is useful for the reader? That is what I'm trying to say. So the stages of ML should be reproducible, and that's where you have to apply it. And what is not machine learning? Artificial intelligence is not machine learning, and rule based inferences are also not machine learning. I'll be discussing about rule based inference with respect to multi parameter optimization. In pharmacology, I remember in the CAD paper, they have a terminology called multi parameter optimization, which we will discuss today based on the rule based. And then let's look at the application of ML in chemistry, how the chemical data is being looked at in data science perspective. In early 2018, CNDN has taken a survey and found that more than 44% of the chemists agree that ML is overhyped or AI is overhyped. But that is back in 2018. Right now, in 2021, I am sure that this would have been reduced dramatically because ML showed its lot of importance. But again, we need data. That is something that we can do it right now. As I started my slide with saying that we are not yet ready for this AI in drug discovery. But what we have to do right now is we have to generate as many as possible data required So it is very important that we also generate possible data so that we will be able to make these ML or AI models viable tomorrow. Unless we have the right data with us, we cannot make sure that these models are accurate. So make sure that right now our agenda as a scientist is to generate as many data available for machine learning. So if you look here, the chemist in this particular survey was 23% was organic chemist and 16% was analytical chemist, and then looking at 10% biochemist. And surprisingly, if you look at the percentage of computational chemists, it's less than around 10 percentage. But now many of the computational chemists started using it. Even in India, we have many groups who are working towards machine learning, which is being very well applied. So ultimately, let's also look into a few other applications where machine learning is being utilized. In my next slide, as I move on, you see the normal workflow of machine learning being used in the synthesization of reactions. So the workflow of chemistry is when you try to synthesize compounds like chemist analysis, like annotating productions using sensors, screening, simulation, etc. And then we have to pre-process this data. Pre-processing or data transformation or data wrangling, this is something very important. We will discuss this during the demo. So you cannot give any data to the machine learning models because this has to be well trained, this has to be well prepared and well transformed. Most of the data that you're getting is not normally distributed. So we have to transform the data by taking its logarithm or taking the factor. And then we can put it into the machine learning techniques like or algorithms like SVM, neural network, decision tree, and many others. And then automatically, when more data is being put into the techniques or models, they will train themselves. And that's where I said new data put in, they get into trained the model. And then based upon the data type, we can say whether this is a classification based model or a regression based model. Now, when you're looking at the chemistry data, this is again taken from public domain. So if you see here a project-oriented data set, you have the design of experiments, you take care of experiments, and you carry out experiments. From that experimental data, you try to generate the model. And as and when the experimental data is being fed, we are trying to generate as many models available. So the modeling part is going continuously when the data is generated from your wet lab or physical experiments. So active learning is happening. And later we are trying to optimize it. So I'll be showing in the demo optimization step also on the ML based. Okay. So today during the demo, we will be looking at how to optimize properties or descriptor properties in order to get the best activity data. And second, how to optimize the chemical structure in order to get less toxicity or better activity. These things will be looked at today's. So using these ML based models, understanding these experimental data, we will be we will be able to come up with new proposals. Okay, these kinds of structures or these kinds of reaction condition will give us a better yield or better purity. These kinds of analysis is possible in machine learning. And another thing is like if you have an in-house data set, for example, when I was working with Professor Allen, he was having his own archive of synthesized molecules, thousands of molecules, right? So it is called in-house database. When they have an in-house database, we can train or we can generate a model based upon successful reaction and failed reaction. When you're doing modeling, it is important we need to have negative as well as positive results. Otherwise, the models cannot predict positive or negative. So don't think that only with the good active you have to do modeling. No, I have seen this earlier also. When they're trying to split between training set and test set for QSR, so some of the scholars try to say that okay, the best ones in the training set and the bad ones in the prediction set. No, you have to have a mix. Bad and good should be there both in the training set. So then we try to understand based on several models. Later, we will be able to understand can we predict what should be the activity in the system based upon the history of the reaction. So SVM models or support vector machines will be able to help with us. Another applications is that simulation, for example, data simulation. I know that there is a software called AIMS, Amsterdam Modeling Suite. They have implemented this called machine learning potential. For example, when you do DFT, you need a good computer. You need more number of CPU cores or RAM because it takes more time for calculation. But this is being replaced by ML based methods, so machine learning methods. And there are many codes available freely in the GitHub also. So that's how we do it, right? I also learned machine learning from GitHub, where many other codes are there. Then we try to take it, we try to optimize it, we make changes, we implement something new, and then come up with a code or a pipeline. So here in the data from simulations, you can see how ML is predicting the energy terms with respect to DFT. ML will be much faster when compared to DFT. But for a smaller system, this is good. For a larger system, we have to wait and see how far the accuracy comes when using machine learning models. When you're looking at neural networks, for example, for a reaction, we want to know what should be the best catalyst, best solvent, best reactant, optimal temperature that neural network to understand or to model it to understand, so that when you give A plus B to C, the model will be able to tell you what are the optimal parameters for this particular reaction. Similarly, we have deep learning algorithms to understand each and every property at different perspective of the molecules. If you look at the center of this particular slide, you can look at the materials design, you can look at the drug design as well as molecular design. Okay. Sir, can you please confirm? I got disconnected in this slide
Yes,
or
sir.
the
You please
previous
continue,
slide?
sir. Continue, sir. Thank you,
Oh, okay. Okay.
sir.
Sorry about that. I think there was some network issue. Anyway, I will just reframe from this. So you have to make sure how to interpret these schematic representations, which is very, very trivial and important when you're going for machine learning models. And finally, what I was trying to tell here, I'll repeat it here because my network got lost. So you have to make sure that you have to apply multiple techniques or multiple algorithms in order to come up with or decide which would be the best model. And today, during the demo, we'll be doing that. We'll be using around 6 or 5 different algorithms to generate the model. So make sure that when you're doing QSR also, make sure that you use multiple methods or multiple algorithms to generate the model, validate them, and report the best one. And make sure you report all these things because it is good to the readers that to know that okay, these types of data or these types of class of compounds or these kinds of distribution of the biological data doesn't fit into this particular algorithm. So it's very important we have an understanding about multiple algorithms in machine learning. Now, how do we transform the chemical data or a chemistry structure data to machine learning algorithms? That is where we try to generate fingerprints, right? So we try to have the chemical structure. A global fingerprints can be generated. These fingerprints are could be bits and binaries, but for the specific region of the molecule, which could be like a functional group or a hydrogen, like these different specific groups. So these can be also looked at where we are trying to understand okay, this specific ones applies to these specific amino acids, and these amino acids are also very important, sorry, the atoms or hydrogens or functional groups. Basically, I want to say functional group, not amino acids. So these will generate a specific fingerprints. So collating all these fingerprints, that's where the global fingerprints comes into the picture. So make sure that we understand the chemical structures are transformed to fingerprints for the machine learning algorithms to understand what are the specific region or what is the selectivity of these specific regions of the compound. Then only we can understand the atom contribution towards activity. For example, we are saying this particular compound is toxic. The machine learning model will be able to tell you based upon the trained understanding will be able to tell you that okay, this particular region is more toxic. That is because they were able to understand that atom contribution towards that toxicity value or toxicity mode. So that's how we are trying to train with the descriptors. So now let's look at a few techniques how to avoid these failures because that's very important. Now, let me tell you, there are hundreds of softwares to help you how to do or how to perform docking QSR. But there are only very few softwares which helps you to understand what is the right way to do docking or what is the right way to do QSR. So there are different perspectives how you can carry out these experiments. Now, this all depends upon the availability of the data also. If you have the 3D structure of proteins and if you have the 3D structure of the ligands, that's where you perform molecular docking. Now, if I ask you what is molecular docking, we all will say key lock hypothesis. Very good. But what is the next level of explanation? I would personally say molecular docking is a process to identify bioactive conformation of your molecule within the receptor. It could be protein or DNA or RNA. So the finding out the optimal binding bioactive conformation is something very important, and the strength of that is being quantified by docking score or binding energies or binding affinities, right? So until unless you have a 3D structure of the protein from X-ray structure or X-ray diffraction, you cannot perform molecular docking. And again, molecular docking is a bit rigid. I mean, making the protein rigid. So we all know that in real life, atoms are in constant motion. So 100% you have to do or perform molecular dynamics. And if you don't perform molecular dynamics, your molecular docking studies cannot be validated computationally. So this is where we have to make sure that just doing molecular docking do not make decisions to purchase a compound and do testing. No, it might not be better than your reference molecule or your standard molecule. So we have to make sure that we do the proper analysis before making decisions. So this is called structure-based drug design. And let's imagine that you have a 3D structure of the protein. You tried all database compounds and tried to do docking and understood, but then you couldn't find the right ligand or the small molecule for your protein. In that case, we try to end up with scaffold hopping or de novo design, which is called fragment-based drug design. And here also there is a catch. Don't think that any software that is predicting these fragments are all synthesizable, even though the algorithms checks the synthetic feasibility. So that's why I used to say fragment-based drug design is best for medicinal chemist or organic chemist. But if you're a biologist, then better have a chemist sitting next to you when you do fragment-based drug design. Let's imagine when you don't have the 3D structure of the protein available and you have only the ligands available or the small molecule data available, including the activity data. In that case, we will try to do pharmacophore, similarity analysis, and QSAR. But be very careful on what kind of data you are using. Let's say pharmacophore. When you're using pharmacophore, your reference pharmacophore map or pharmacophore features should be from a co-crystal ligand or from an X-ray crystal structure ligand conformation, bioactive conformation. If that is not there being used as a reference, there's no point of doing pharmacophore screening. If it is a QSR, let's say 2D and 3D, everybody is fond of doing 3D QSR only, but ultimately most of them are not doing it in the right way. What we usually do, we generate 3D structures from the 2D by using molecular mechanics or semi-empirical methods, and from that 3D structure we generate the descriptors and model it. This is really good if you're trying to build up a model for physical or chemical properties. But when we are talking about biological activity, we already know that the conformation of or a geometry of a compound in a stable atmospheric pressure or on a stable room temperature is different from the conformation or the geometry when it binds to or forms a complex with a protein. It is different. So when you're doing QSA for bioactivity data, are you considering the room temperature stable conformation or the bioactive conformation bound to the protein? I think you're able to imagine it. Sorry, I cannot get your audio feedback. I hope you're able to understand. So ultimately, when you're taking the 3D structures for calculating the molecular descriptors for QSAR, make sure you are taking it either from the pharmacophore screened conformation or from the molecular docked poses. This will give you much more relevant descriptors, 3D descriptors, especially 3D descriptors to correlate between your bioactivity prediction. So 3D means everybody is fond of, but make sure you are using the right 3D structure to generate 3D QSI models. And that's where we call it as ligand-based drug design. So for structure-based drug design and fragment-based drug design, we have some software called as CSAR, which does a better job. And then going for ligand-based design or ligand-based drug design, there is something called as star drop, which I'll be giving you a short demo after this presentation. And when you do not know the right target or when you do not know which could be the best molecule, that's where we do a high throughput screening. And that's what we call it as combinatorial drug design. Okay. Before we move forward, is there any questions or comments? No. Okay. I don't see any questions. So if you have any questions, please put it in the chat window. I will look in now and then so that I can answer them. Okay. Thank you. So moving forward, what is molecular docking? Looking at molecular docking, earlier days we used to say key lock hypothesis, but that is only half truth even in 2021 or even 2020 also, because now we are talking about drug repositioning or drug repurposing. So how can we say one lock, one key is for one lock? One key could be for multiple locks also, right? So we have to see how this could be redefined. Now, why we do molecular docking? We do molecular docking to understand the bioactive conformation, right? As well as we want to know, look at where the ligand will bind, how will it bind, how strong it could bind, what is the role of solvation or desolvation, what makes a ligand binds to the receptor better than the others, translation and rotation of ligands, including torsions. So the bioactive conformation should form a co-crystal also. How can we quantify understanding the co-crystallization of a ligand? And that is through torsions, torsion energies and torsion bonds. We try to understand statistically. Now, looking at the right hand side of this particular slide, this picture is taken from Enzyme Methodology book, where we are trying to understand the catalytic function, regulatory mechanism, cleft detection, geometry analysis, flexibility, conservation, surface characteristics, and interaction energy. What we are trying to understand here is that before doing molecular docking itself, you need to have a clear idea on what kind of amino acids you have to focus on interaction. Like whether you are targeting a catalytic site or a motif site or a specific functional site, you have to understand. It is not the other way around. Okay, my compounds docked and interacting with these amino acids, these are the amino acids important. No. So, in order to have a mechanistic driven drug design or understanding the mechanism, you have to make sure the interactions or the amino acids are also biochemically justified. So, make sure whether you are looking at the regulatory mechanism, whether you are targeting the active site or the allosteric site. When you're looking at the flexibility, whether it is apo site or substrate bound site. So these kinds of understanding is required in order to say whether your compound is agonist or antagonist. No softwares can tell you whether your compound is agonist or antagonist until unless you understand this mechanism part, right? When you're coming to conservation studies, make sure you look at selectivity, specificity, and sensitivity. I'll give a small example. Let's take a target for HIV reverse transcriptase type 1. How confident are we that our compound is going and interacting or docking only with that particular protein target, not with any other protein targets? We might say, I am confident, but then where is the data? Because we wanted to see what are the other proteins which is having similar binding pocket or binding site of the HIV reverse transcriptase type 1, and so that you go and dock all these compounds in those all proteins and then tell that your compound is very much selective or specific to this particular HIV reverse transcriptase type 1. But most of the time, we select a target, we do the docking, report the data, finish the job. That is not the right way. So selectivity, specificity, and sensitivity has to be reported when you're doing docking analysis. Similarly, in wet lab studies, if you have funding to carry out that particular assays. So what is that is important here in ML? That is what is coming to, I'm coming to that point. Because ML, if you say, yeah, for this particular target, I got these are the binding energies, and I'll generate a model or I'll generate a training set. Later, they don't look at specificity, selectivity, and sensitivity, right? So the selectivity, sensitivity, and all those things can be a feature, can be a variable that to be considered to make decisions for the model. But are we really giving these data? No. That's why I said we are still not yet ready with AI in drug discovery because we have very sparse data, right? Now, that's where, so far we were talking about design. Now we are getting into discovery. When you're talking about drug discovery, we have to transform from the hit identification to lead. Hit identification is just a very minimal part of the whole drug discovery project. Because in hit identification, that's what you're trying to do, molecular docking, and then you will try to do bioassay studies or potency studies. That's where you try to identify the hits. Now, when you're doing molecular docking, don't think that you are ending up selecting one or two molecules. You have to at least select 50 to 100 molecules. Now, you might say, no, we don't have funding for that. You have to proactively ask for 200 molecules testing. And so that the funding agency can give you money for at least 30 compounds or 50 compounds. So make sure that your pipeline and your protocol is very well diversified, and ultimately you don't get the good results as expected. And that is because diversity is not there. So when you want to transform from hit to lead, you have to make sure that you have to understand what are the pharmacokinetic and pharmacodynamic properties. So, if you are not able to achieve this balance of properties as early as possible, failures are that's what you look at. So, fail fast, fail cheap is mantra in the industry, and including only when confident you move forward. Another challenge is that avoid missed opportunities. Very important. I don't know if you agree, you can put it in the chat that do you use Lipin's rule of five to filter the database compounds before doing or before performing docking? And if you do, please don't do that. Because while doing hit identification, you need to have a better room or chemical space. I don't say you need not to filter, you should filter, considering the size of the ligand or certain ring parameters and others, because you better know what is the volume or the size of your pocket, protein pocket. So it's to save time, it's better to select something very proactively, what could be the best ligand that could fit into that pocket. So based on that, you can try to filter them, not just based upon Lipin's rule of five. I didn't say Lipin's rule of five not to be used. Yes, it can be used at a later stage, but not before docking studies. So hit identification should have a better diversity and chemical space in order for further optimization. So that's where we look at the data overload challenges. There are many compounds. Let's imagine, I'll make it simplified. Let's imagine there are 50 different compounds we selected. And then we carried out 10 different experimental analysis. So from the 50 compounds, I want to select only 5 compounds, right? When I'm selecting these 5 compounds, I want to make sure that this 5 compounds satisfies all the 10 experimental data, right? So among the 50 compounds, I want to select the best 5, considering all the 10 experiments data. You might think whether can I take an average or whether I will do it one by one. Let's see what can be done. So initially, when you have a lot of data, in silico, in vitro, in vivo data, usually we do it one by one or one after the other. But you also have to make sure that we have to prioritize things. Potency or activity data is one of the top most important parameter in the drug discovery area. And then we have toxicity. So you have to set the importance level for these parameters. And next is uncertainty, error value. If a property is having more error, even though that is a most important property also, don't consider them as most important because it has more uncertainty there. When there are more errors and further you are using that to generate a model for and using that particular data, you are adding up more error to the model also. So it's better to understand uncertainty and then define the importance. And finally, uncertainty and okay, I can see that some audio problem that you're reporting. Is it okay with everyone? Can you put it in the comment? Okay. the audio is fine. Just the audio is not continuous. Is it still breaking?
Audio is not continuous. Somebody is telling, sir.
Okay.
Audio is not continuous. There's some network issue is there. That's why, sir.
But are you able to hear me properly, sir? Oh, now it is okay. Okay. Thank you.
Now okay, sir. Thank you, sir.
Yeah.
Thank you, sir.
Sorry about that. I'm at my home, so I don't know whether the network is bad. Yeah. Sorry about that. I'll take care of this hereafter. I'll look into see whether it's properly connected. Okay. So looking forward to QSAR, we are trying to understand. We want to extract the properties from the molecular structure, and the molecular structure can be 2D or 3D. I already told you how 3D to be considered. I'm not going to repeat that here. From the molecular structures, we have to extract molecular properties. And these molecular properties comes are called as molecular descriptors. So these molecular descriptors are something very important that we are looking at, because these descriptors can be thousands and many others. But make sure that does it have any physical meaning? Simply generating lot of descriptors doesn't make any sense. There should be meaningful descriptors coming in. That is something very important. So wisely choose your descriptors. And then the corresponding experimental data from the molecular structures will be as a Y response variable. And that is your activity data. And then we are trying to use some molecular descriptors and the response variable, that is experimental data, to correlate and generate the model. While you're generating the model, you can use either statistical methods or machine learning methods. Statistical methods, we mostly, even my favorite is also linear regression method. But unfortunately, linear regression method has a drawback of overfitting issues as well as false positives. So it is highly recommended linear regression to be used on a homogeneous data set or for a local QSR, not for a diverse or a global QSR. Or to overcome this, the other statistical methods are partially squash. Okay. Now, coming to machine learning, we can use random forest, we can use decision tree, we can use this Gaussian process regression method, or even deep learning method if you have enough data available. So this is where we have to make decisions. So literally, as I told you, you have to generate the model using all the techniques, and ultimately, when you're trying to validate them, make sure which one you choose. And that's the time where you choose which is the best model that I have to report or deploy. So don't think that just using Bayesian model or just using random forest, I have to come up with a model. No, you have to come up with multiple models using multiple approaches, right? And once you validate, you will be able to predict the activity property based upon whether it is chemical or physical or biomedical. But keep in mind, any of these QSR models that you're generating, you have to report the standard deviation or the standard error. And that is also very important. Without a standard deviation or the error, QSR properties, or just a minute. Yeah, QSR properties or QSR prediction is not appropriate. I can see some of the participants are asking for the slides. As I told you, the slides are already available in slideshare.net/GiriBio. All these slides are available. Okay, so now what are the steps involved in the QSR? You need to have molecular structure, you have to calculate descriptors, you have to classify the data set according like training set, test set, and validation set. Then you have to select the feature selection, which descriptors is the most important one, and then you generate different models and then you validate it. Now, after when you come into the validation, it's also important to look at the domain of applicability. I used to tell this example every time I present. Let's imagine you have a single structure and you used 10 different softwares to predict solubility or ADME properties. Let the 10 softwares be paid, free software, or even online web servers. Now the question comes, all 10 gave different results. Which one should I choose the best ones? The answer is very simple. One, which other models gave the same answers? Number two, very, very important is whether what is the standard deviation or the error predicted for this prediction of your molecule. So for your molecule, the 10 of 10 softwares would have predicted the values of solubility or ADME properties, which all tools was able to give you standard deviation or error value. What it tries to imply is the domain of applicability. Let me explain that. Imagine these orange colored dots are your training structures or the structures in your training set, chemical space. The blue line is telling the boundary of the chemical space. That boundary of chemical space is called applicability domain or domain of applicability. When you're bringing in your structure, like a green color star, if that structure is fitting within the applicability domain, or is it having a similar chemical structure within the training set or same class of compounds, the standard deviation will be minimal. That is more reliable prediction. If the standard deviation is slightly higher, that means it's outside the applicability domain. And if the standard deviation is so far, that means it's a bigger number of standard deviation. That tells you your prediction or your structure is not within the applicability domain of the training set because your structure is entirely different from the structures used in the class of compounds in the training set. So until unless you have an understanding about standard deviation or within the applicability or outside the applicability domain, you cannot say the prediction by software X or Y is accurate. Whether you pay $1 million for a software, it doesn't matter. It's about whether the applicability domain is known to you for the prediction ability or not. When you're having challenges, of course, machine learning is not that easy. You require large and good amount of data. So there are challenges on applying the deep learning. One of the biggest challenge, as I told you earlier from the beginning, is was sparse data. We don't have too much data available. So we try to do data imputation. And another thing is that we, due to human errors, experimental outliers are also there. So, and another error is that if you don't repeat your experiments, then we don't have a standard deviation, right? Or we don't have an error reported. So that is called very uncertain. So if you're running your experiments only just for once, then we are very uncertain whether that particular reported value is correct or not. And similarly, as I told you, imputation. So you have different descriptors and coming up with prediction. Similarly, we have only few assays for certain compounds, but we can fill in that gaps with the process of imputation. So we need to have a much more situation with the multidisciplinary aspects where chemists can talk to each other, to modelers and informatician, so that they all can work together for much more productive result. Medicinal chemist or organic chemist tries to understand how the docking results to be looked at in order to make new analogs. Which region of the compound has to be modified? The computational modelers will be able to help medicinal chemists to understand where are the possible optimization could be taken care, and is this reported earlier or not. Informaticians will be able to implement existing data which is generated by the chemist to generate a model and then come up with a prediction or a proposal whether the proposed reaction could happen or not. Now, I don't say it's 100% accurate, but at least it can predict based upon the history of the data that you have given. So we need a multi or interdisciplinary combination for each of these projects. Now, if you ask me what is the success story of the computer-aided drug discovery, this is the success story. You have plenty of compounds which is in the preclinical and clinical trials, and I have taken from these two reviews, and it's a very good blog spot or a blog which is Practical Fragments, which talks about fragment-based drug design. So this gives a better idea about how these compounds are being used, how docking, pharmacophore, and many other optimization techniques being used to come up with these molecules. Now, so far we discussed about what is machine learning, how chemistry data is applied in data science, how to avoid the failures, what all things to be considered, and then what to do next with AI. This is not my personal view. This is a view that is taken from a AI consortium. Even Europe recently compiled a AI policy. It's very important because we need to make sure that humanity is also being preserved and there's no danger to the human jobs or humanitarian. So, artificial intelligence, they are looking forward to the next coming years. We have to be so careful and we have to come up with the models very carefully. This is the reason why I told you earlier also. A very famous company stopped their robotics project just because the robot themselves started to talk to each other between the two robots, right? Usually we humans only do that, right? But here the robots started talking to each other. That is not good. And they wanted to further research how they spoke, how come they try to understand each other like humans, which is alarming for them. So they stopped that project. So this is similarly in par with this consortium also, where they are looking at whether jobs are at risk or humanity at risk, and they still don't know. Of course, Europe came up with the AI policy. Similarly, I am sure other countries also will come up with AI policy to have all the restrictions where it to be used, where not to be used, all those things. So to conclude with my presentation part, before we go into the demo, make sure that you initially consider 25% importance to the score or quality and 75% to the diversity of the molecule to have a diverse chemical space. And when you're reducing the number of molecules at the end, when you reach to a lead level, hit to lead, at that point, consider more importance to the score that is or quality that is 75% and less percentage to the diversity because that's where the optimization would have happened. Consider enrichment factors in order to calculate the quality of the virtual screening. Good synergetics between human expertise and computational tools is very important. Don't think that computational tools can make wonders until unless you give right information to the computational tools. Try to avoid missed opportunities without considering the right strategy or the right rules or filters. Understand the significance of the parameters means try to use the right descriptor for your activity rather than simply based upon R square and statistics. We need to have a balance of both. Evaluate and decide the tools or approaches. Don't simply think that today I'm going to talk about StarDrop or today I'm going to talk about Rapid Miner. Don't think that that is your best software that you have to use throughout your life. No, it might be working for me the best. So make sure that you have to try yourself. All the software is paid free. Everything you can try yourself at least during the evaluation. And then try to see how it fits into your data, whether it predicts better than the others. Check the reliability of the data that is being used. That's how you have to choose the right algorithm or the right tool. So you have to evaluate and decide and check the reliability of the data that is being used. So that's all from me for the presentation part. Now we will go to demo, but if you'd like to have a quick break or a quick pause, you can do that. And we will go to the demo. And if you'd like to have a quick break or a quick pause, you can do that. And we will go to the demo. But if you'd like to have a quick break or a quick pause, you can do that. And we will go to the demo. And if you'd like to have a quick break or a quick pause, you can do that. Hi, sir. Can we start?
Yes, sir, we can start, sir, now.
Okay.
Okay, sir.
So, yep. So, thank you all for still staying with us patiently. So, if you have any questions, quickly put it in the comments. I'll in between look into the comments because we don't have an audio provision here. So, make sure that you clear your doubts and go. So, this is a software which I was mentioning about Star Drop, and it is mainly for ADME prediction, human toxicity prediction, and it will be also giving you all information about the statistics, standard deviation, multi-parameter optimization that could be performed. As I said, multiple properties can be combined together and using a rule-based inferences for probabilistic scoring to understand how far all these properties can be considered together in order to choose the best compound or remove the bad molecules from our data set. And this is a feature called as a glowing molecule, which tries to helps us to understand which region of the molecule is significantly or having significance in solubility or Herg liability and all. Now, this is what I was explaining in the presentation about atom contribution. So the descriptors or the fingerprints try to understand the atom contribution and try to give you this information. And then we have something called as P450, which tries to understand metabolic stability. For example, it tries to understand which region of the molecule is unstable, and then where it could be, for example, break down or form a metabolite. So once it reaches your target, it might not be reaching the way you have did the docking, but it could have formed a metabolite before it reaches the target. And then if you wanted to design new molecules based upon the property, again, retrosynthesis, all those things can be carried out, including QSCR, which is machine learning based QSCR, also can be carried out. Now, I'll be telling the alternates, but this is a paid commercial software. Like tomorrow, also you have one more session from another team. So similarly, this is more focused on lead optimization, not only on the structure based. This is more of a ligand based lead optimization part that is coming up, including pharmacophore analysis. Now, let me give you a quick analysis here, and then we will quickly go to RapidMiner to talk about machine learning models and others. So here in QSR also, we do have machine learning models like Gaussian process regression methods and others. But let us do an analysis on the multi-parameter optimization, because anyway, this is there in your MPharm curriculum also, this will be useful. Okay, let me give you an example here. So this is the activity data for each of the compounds. Now, how to take the input to this software is basically SDF files. You require SDF files, that's all. So I have the structures in 2D and I have the activity data. And if you have the names of the respective structures, that would be nice. Now, based on the activity data, this particular structure, let me show you that here. Yeah, this particular structure is having the best activity, which is 8.142. Now, this is a hit molecule now. I cannot say this is a lead molecule until unless it satisfies all the other ADME properties also. So here I will be able to predict the ADME properties like log S is aqueous solubility, log D is again the model predicting the log D dissociation, log P partition coefficient, erg liabilities like cardiotoxicity, blood-brain barrier, human intestinal absorption, plasma glycoprotein 2D6 and 2C9 are the isoforms of P450, cytochrome P450, then plasma protein binding categories PPB9. So all these will be predicted, and then we will be able to combine the bioactivity data, including the ADME properties together in order to score it from 0 to 1. So probabilistic scoring is between 0 to 1. But let me explain in a simple way how this importance level is given. Let's imagine, I hope I'm still shared, yes. So let's imagine a Lipinski rule of five, where I'll just pick up the example molecular weight. We were being told that any compound between 0 to 500 molecular weight is good. But we already know that the compounds which are FDA approved, they also have molecular weight above 800. So when you filter the compounds, saying that based on Lipinski rule, I'm removing the compounds because the compounds with molecular weight greater than 500, I'll be removing. But that is what is called as missed opportunities. We do not want to do that. Rather, what we will do is, in the considering the molecular weight, we will say, for example, I will add molecular weight and explain it. Yeah, molecular weight, add rule. So I'll just drag it here. So I'll say molecular weight less than or equal to 500 is acceptable. Okay. But I also wanted to consider other compounds also. For that, I'm going to this particular place. So this is where the multi-parameter optimization comes into the picture when you're defining the score for each of the properties. So any values from 0 to 500, I give a score of 1, and any value between 500, or let me add insert a value. Yeah, I'm trying to say now any value between 600 to 700. I'm giving a score of zero point 0.5. So 600 to 700 is 0.5, and anything, or I can again make between 500 to 600, I'll say 0.7. Right? So again, I'm saying, okay, between 700 to 800 molecular weight, I'm saying to give it 0.3. And anything above 800, I can change it to 0.1. Okay. What it tries to tell you here is, I'm not leaving any compounds anything above 500 molecular weight. I'm still considering them, but I give a less weightage or less importance to it, right? But in my this particular data set, I don't have any compounds with greater than 500 or 600, right? If it was there, there could be red colors coming in, but here it's all green color. So this, so if you normally used Lipinski rule of five, all the compounds above 500 will be completely removed from your data set. But in this condition, we are trying to give them a less importance, means lesser score. The score is between 0 to 1. So lesser score is given so that it will not come to like a more importance level. Okay. So I'll just delete this because I have already prepared this data for explaining something different. So based upon these values and desired value and importance, what I explained to you right now, I predicted the values here. These are the scores I am having. So let me score yeah, sort the score based upon highest one. So this particular compound is having the highest score of 0.41, but its bioactive or the activity data is only 7.52. Let me see if you have any questions so far. Okay, no questions. Okay, so yeah, so basically we are trying to understand the activity data. So here the activity data is 7.52. Hope you all remember the best active data or the compound was having somewhere around this one, 8.142. 8.142 is the best topmost compound in this hit molecules. But later we could see when we added ADME properties, we could see this compound with 7.52 is the best. Let's analyze what happened. Why did the software recommend this software to be the best when compared to the other one, which is even though having a better activity? For that, I'll go to the glowing molecule. So we already know that our activity data is most important one. But here you can see the activity data is lower here. The other compound is having the highest one, but still it did not come up. Let's see what are the other issues. So looking at the solubility, you can see this compound with 7.52 activity is 4.185, the aqueous solubility. But the most active compound, this particular compound, the solubility is only 3.8, and that was the difference. Not only solubility, there are other factors also. Let's look at the Hurg liability, cardiotoxicity. We have 5.6 here. When you go to the topmost one, which is suggested by the software based on rule-based inferences, here the Hurg liability is only 4.5. It is less. So here in the scoring, we already said if the Hurg is less than 5, it should be given more priority. But the best active compound was having a Hurg greater than 5. I hope now you are able to understand now. Even though the activity is best, until unless other properties are having optimal balance, you cannot find a good compound or a good lead molecule. Now let's look at the visualization part, because I was telling you schematic representation is something very important in data analysis. So when I look at the schematic representation, this is called snake plot. So on the y-axis, it is the score ranging from 0 to 1. And on the x-axis is the compounds. The colored circles are the each of the compounds score values. Now, each of this prediction also has standard deviation or error being predicted. Now, which is evident here is that I'm very confident to select these compounds and remove it from my dataset, because they are no way coming closer to my prediction or expectations of balanced properties. So that's why I'm completely taking away from them. So this is something very important we have to look at. So that's where we understand whether we want to do computational studies based upon choosing, like our aim is to choose the best molecule, or our aim is to remove the non-promising compounds from our data set. So this is exactly that we are looking at how things move forward. So make sure that how the perspective that we're looking at analysis, this is something very we have to look at on the different aspects of computational studies. Many of us, including me, we believe that we use computational tools to choose the best molecules. But let me correct you there. The best molecules comes only after wet lab validations. Using computational tools, it gives you idea, it gives you decisions, it helps you to make decisions. Don't think that computational tools are here to replace your wet lab experiments. If someone is telling you that the computational tools can do that, sorry, it's your risk to believe it or not. But for me, computational tools helps me to make decisions whether to choose this compound or not. But I will never say this is the best compound. So these kinds of analysis is required, implementing statistics, implementing machine learning to the decision making criteria. All right. So, and ultimately, I want to show one more example and then we will move forward. One more example is that this, let's imagine this, our compound, the best active compound, the 20th one, it was showing the toxicity bit problem in certain regions. Let's see how we can quickly reduce it. If I am a chemist, if you think that you are an organic chemist, you know what you're doing, you make modifications and you want to see how quickly these things can happen. So what I'm going to do is that I will make this convert to carboxylic acid. Okay, I'll make a double bond and I will make it hydroxyl, so carboxylic acid. You could see the Hurg liability from 5.6 reduced to 4.032. Now there is a catch. Let me show you that also. Now there is a catch. What is the catch? I do not know whether this can be synthesized or not. So in order to know whether this can be synthesized or not, I can do a retrosynthesis pathway predictor. Many of the chemists in this participant list might be knowing SciFinder or Reaxys. These two tools try to do a backward search to understand whether these compounds can be synthesized or not based upon the existing literature data. Similarly, in StarDrop, there is a freely available feature for academics that is called IC where you will be able to look at the synthetic assessment. When I made a modifications on the carboxylic acid, it will be able to search online server based upon machine learning algorithms. It will be able to come back and tell you this particular structure is similar to your structure, and this is a particular yield that you can achieve from this structure, and what are the literature references that could come up. So these are all very important factors that we have to see how these computational tools or machine learning tools are able to help the chemist or pharmacologist or drug discovery scientist to make decisions. Now, once this literature comes up, it is very easy for a chemist to design his routes or design which starting reagent, where he have to start, all those things. So these are helping tools, assisting tools. That's why we call it as computer aided drug design. It's giving you an aid. It's not replacing your wet lab. So we still need to have the synergetics between that, the expertise and putting it there. So based upon the internet speed and the complexity of the structure, it might take some time. Let it do. Once it finishes, we'll come back to this. I will show you the results. Okay, now another mode is you are not a chemist, you are a modeler, and you don't know really something about synthetic chemist, but you still wanted to work on this compound and see whether we can reduce the toxicity so that the activity enriched compound will be much better. For that, we have four different kinds of approaches. One is called chemistry transformation, another is matched series analysis, reaction based library enumeration, and scaffold based library enumeration. So these are widely being used in pharma industry. I don't think that you cannot use it. You can also use it everywhere the science is saying. So when you're looking to the chemistry transformations, so we are trying to define which region to be made it stable. So I will say this region I do not want to make any changes, but the other regions I want to make changes. And then the software will be able to use certain linking and breaking rules in organic chemistry, try to understand what are the linker, what are the possible substitution reactions, what are the possible replacements or transformation that could be. It is not replacement, it's called transformation. Bioisostereochemical replacement is something different. So here, the good thing is that it will come up with the literature references. So it will tell you what are the possible transformation based upon the selection of the bond that I have given, and it will be able to tell you. So let me see by the time. Yeah, the results for the retrosynthesis has come here. So here it tells you this particular structure. When I click on the root one, it tries to load the specific references. I have three different literatures, and it is saying there is a similarity of 60% with certain condition, and this is from Jack's journal. Most of them are from Jack's. Now, if you think that I do not know how to make this compound, I want to know the retrosynthesis pathway for this compound, then you click on this plus button. For that compound, it will be again generating the possible routes. So I'll click on the route 11, and then it will be able to generate the possible literature. So this is very much handy for a chemist when you do a lead optimization. Ultimately, at least through a idea making process, you will be able to understand what are the possible retrosynthesis or what are the possible synthesis already being reported with the starting reagent information. Okay, I'm not going deeper into that. I have to still cover the machine learning aspect, so I'll be just quitting this now and moving forward. So this we were doing a medicinal transformation in StarDrop. So here what here the software is trying to tell us, there is an isopropyl group in your compound. So isopropyl can be transformed to pyrrole. Now how to do that? If you click on manage, there is a journal or a literature from Journal of Medicinal Chemistry journal. They have already reported how to transform propyl to pyrrole, where they have already reported the bioactivity as anti-ischemic or cardioprotective, right? So these kinds of information you will get from the transformations, and only the possible transformations are given. Not everything is here, right? Another one is ethyl to acycloxymethyl, and if I manage, I can see these are literatures where they have already reported about these transformations, and this is what is called lead optimization. Now, this is something very important. When you do replacements, it is not simply that based on a database, simply make any replacement. I'm sure that all of you are aware of toxic groups, toxin groups. When you replace, you have to make sure that certain groups are not coming into the drug molecule, like disulfides, thiols, thiocyanate, thioesters, cyanamides, right? And similarly, many others, primary alcohol, hydroxylamine, acetal, or nitrogen functionals like amides, sulfonamides, certain things. So we have to have a clear picture what kind of transformations we don't require based upon the toxic groups are already reported. If you want to know about that, you just mouse over on top of this particular list. You will be able to get the literature references so that you will be able to identify that. And then now I don't want to simply make replacements. I am doing it with a specific reason. My reason was I want to reduce the hERG liability of this compound without affecting my activity. So I'm saying I need a low hERG liability. So let me search for hERG property, and I said hERG, and I click next and finish. Few more options can be done for generating large amount of compounds, but for a demo purpose to make it faster, I did not give that stringent criteria. Now, this is my compound which I selected to design my transformation. And here I have a hERG liability of 5.6, which is high, which we already know. This is the most active compound, but still it's having toxicity. Now we have to come up with a different optimized compound so that it will have a less cardiotoxicity. So I was able to come up with few compounds which is having lesser cardiotoxicity, which is 4.1 when compared to the parent molecule, which is 5.6. Now let's see what is this. So here it is trying to tell us phenylalanine to pori oh yeah porphyrinyl phenylalanine. Of course, it's a very large replacement. It is being reported in this particular paper. And let's go to a next one, which is a little more simple. So one is hydroxyl to carboxyl. Yeah, of course, zanamivir. We know about this drug, zanamivir. It's a antiviral drug. And similarly, we can go with another one. Let's see about, yeah, sorry, that is a repetition at a different position. And this compound, guanidine to alkyl, right? We have a guanidine group here, and that's getting transformed to alkyl. And then we have reported in JMET Chem. So these compounds are 100% synthesizable because they are already reported in the literature. So these medicinal transformations helps you to make optimizations based upon specific property. And again, you go back to the multi-parameter optimization to understand whether the activity is good. You can predict it and then synthesize it and test it. So it is an iterative process when you do lead optimization. It's not a single time process. And that's where it is taking time for a drug coming into the market, even to the clinical trial, because pharma industry has to do a lot of optimization before getting into the clinical trial. So I'm sure that all of us will thrive to get into more data into the publication so that the commercialization could happen in much better ideation. So on this context, do you have any questions? Uh, I don't see any questions so far. Oh, the software is Stardrop, Mahindra Pratap Swain. This is called Stardrop, Stardrop. If you have any other questions, you can post in the chat, live chat window. I will be able to read it. Unfortunately, it is not open access. Of course, for teaching, it will be free, but not for research. You have to purchase the license like any other software. But the next demo, what I'm going to show you in machine learning, it's an open access. It's free for academics. But do you have any other specific questions on optimization? I know many of us might not be doing using a software the optimization, but it's already time too late for that. We have to start doing those kinds of optimizations. For docking studies, it's a very well known. It is we have Autodoc, we have Esmina, Vina. These are all freely available docking tools available. Yeah, they are all pretty good also. So Mahendra Pratap Swain, open software or open source software. For the optimization part, unfortunately, we do not have many open sources software available. Most of the time for database management, chem informatics, for descriptor calculation, QSR, of course, we do have softwares open source available, but not that for lead optimization. We don't have much as of known to me. I don't have. Okay, so I don't see any other peculiar questions. So we will move forward with our machine learning model. So let me show you a little bit more information of what data I'm going to use. So I'm going to use QSAR bioconcentration classes data set. Okay, this has around 779 molecules. Now, in order to talk about this, I have taken this from a paper published in 2016. So it is a manually curated 779 chemicals, which was used to determine the mechanism of bioconcentration, which is to predict a chemical mainly stored within lipid tissues, whether it has additional storage sites like proteins, or is it metabolized or eliminated. So the particular paper they have used 75% is to 25% for training set is to test set. But here I did not consider that. I did it myself on generating the different models. Log P is also being used as one of the descriptor along with few other descriptors, including number of hydrogens and number of nitrogens and few other bond formations between carbon, nitrogen, and carbon, oxygen. So this is the overview of the dataset. Now, and this is a software called as a Rapid Miner, or you can use Knime, Knime. So here, just for the ease, I'm using Rapid Miner, which is again free for academics. You need not pay anything. So I have loaded the CSV file directly into the Rapid Miner. In order to load the data, you can go to load data, or you can go to file and then say import data. So if it is a CSV file, it will load like this. This is how the CSV file gets loaded. Now you can do transformation. I already told you if there is something that has to be changed. For example, this is a concentration factor. Now this I have already taken the log value. Since I have taken the log value, let me see the charts. And I need a bar column. Yeah, step area. On the x-axis, I will keep log BCF. Yes, and okay. I don't want all the data. I need only the log BCF. Yes. So here it is showing the, no, I don't want step. Let me show you the step line. Yeah. Histogram. This is the best. So, so we have different visualization charts available within RapidMiner. So what I wanted to show you here was I already took a logarithmic value of BCF, that is bioconcentration factor, to get it normally distributed. If I did not take a log, it was not normally distributed. There was ups and downs. So it is very important that data transformation is being taken care the first time when you consider it for QSA. So don't think that simply loading the data and directly you can go for modeling. Another thing is that you have to make sure what kind of data or data type you are defining. For example, this one is a set. So already the author has given this some of the training set and test set. And moving forward, here we have class of compounds like one, two, three classes are there. So here it is still denoted as number, but I know this is not a number. I have to transform it to change the type to categorical. So I can change here. So you can see change type, change to category. So apply so that this particular class now change to category one, two, three. Some of the compounds are in the category one, some compounds in category 2, and some compounds are category 3. I told you what are those one, 2, 3. Here it is mentioned in the when I was explaining to you. One is mainly stored within lipid tissues, two which has additional storage sites, and three is a metabolic or it's metabolized or eliminated. So those kinds of data is there. And then these are the descriptors, molecular descriptors. And these descriptors in machine learning, we call it as features or feature selection, right? And these smiles and cas number and smiles is called as metadata. Metadata means some reference. It refers to a structure or a name or identifier. So this is called a metadata. Let it that be there. And we have the set of categories. So if you commit the transformation, then only it will accept whatever modifications you made here with the class of compounds. Okay. If you want to add a new column, that can be done. If you want to sample or sort it out, all those things can be done in the turbo prep. So turbo prep is a module which takes care of the transformation and preparation of your data. And you can also pivot the data to look at the sorting and merging data can be done, something similar to Excel sheet. Or you can clean the data based upon missing data and other things. Once the data is being prepared, that's where then we go to auto model. So if you are not too expert in ML machine learning or not too expert in using rapid miner, I would recommend to go for auto model. What auto model does it automatically tries to identify what all algorithms are required in order to generate the models. Once the turbo prep is finished, you can actually send this data to auto model once it is all committed. Once the model will be generated, it will come like this. So I have already the data that is being loaded into it, or if you want to import a new data, you can do this. So if you're importing a CSV file directly to auto model, that is also absolutely fine. Do you have any questions so far? I'll go slowly. Vipul Mahendra for QSAR, as I had also informed last time, you can generate descriptors using PADL, right? And then you can also generate the model using rapid miner, as I'm showing you here. So QSAR doesn't mean that it is a single step. It has optimization, it has descriptor calculation, it has data preparation, and then modeling. So that's what we are doing. So after the descriptor being calculated from PADL or StarDrop or any other tool, you can bring it either model it within StarDrop or you can model using rapid miner also. And then I click next. Now it is asking what we wanted to do with this data. So I already know that I want to predict, I want to generate a model for predicting the bioconcentration factor. So I'll just select the log BCF. So automatically the software identified that we want to predict the value of a column. Then next. So that is going to be our target. So bioactivity data, what you want to model, which is called as a response variable or Y variable in statistics, that is called as a target in machine learning. Whatever you're having the variables for statistics like descriptors or molecular descriptors, they are called features in machine learning. Okay. Now we have this data. We already saw this particular histogram where it's normally distributed on the logarithmic of bioconcentration factor. If you want to transform them into classification, or if you want to change them, or again further transform the data, still we can do at this step. I'm not doing this because I know I want the log values here, nothing else. I go to the next step. Now it is trying to tell you which are the features or which are the descriptors good that to be used for your modeling purpose, right? So if you look at the correlation, so if you keep your mouse over here, you can see this particular property or descriptor is having 22.2 percentage of correlation with your bioconcentration factor with a stability of 37.3, but there are some missing data, about 3.98 are missing data. Now coming to log P, you could see there is a 63.7 percentage, 4 percentage of correlation between log P and the bioconcentration factor. And there is also missing data of 3.98. So this tells you the feature selection, which descriptor is more important than the other. So based on this, I will be able to say I don't want set because I know it is training set and test set what they have mentioned. I don't want them to be a descriptor. And also I do not want a class to be a descriptor. Of course, we can consider at a later stage where 1, 2, 3 are the different class of compounds. So I will select only these descriptors, which is already known to me. These are the having different importance or the features having good correlation with the bioconcentration factor. And then I go to next. Now, this is the place where I was trying to tell you from the beginning, we have to use different models. Don't think that we have to use only linear model or only deep learning or only decision tree or only random forest. Make sure you choose multiple models at the same time and then do this model generation. So you can use images, you can use text, you can use numerical, you can use classification data or continuous data, any data. Automatically RapidMiner will be able to suggest you what are the acceptable models for this kind of data that you have given. Here, I'm going to choose all of these models. Okay, I have already done this, but I'm redoing it for the demo purpose. So I did this already generalized linear model already generated the data. So let me see what is the correlation. The correlation between the actual experimental log bioconcentration factor versus the predicted bioconcentration factor using these models, you can see from the different techniques or different algorithms are different. Generalized linear model is giving around 84% correlation. Deep learning is giving 87%. Decision tree is giving 85%. Random forest is giving 89%. The others we have to see. On the top, you can see the correlation graph, and on the right hand side you can see how much time it took for a model generation. That is also important when you have a large data set. This is a very small data set, you don't need to worry about time, but when you have a very huge data set, time is also something very important. And on the top of this correlation you can see there is a error bar which is also given. So you can see the error bar is reducing as if the correlation is increasing, and the standard deviation is also given here. This is exactly what I was trying to tell you from the beginning. Any software predicting ADME properties, toxicity, or QSR based activity prediction, make sure it has standard deviation. If it does not have standard deviation, it's uncertain whether this result is good or bad. Here I can, by seeing this, I could say that random forest is having the least standard deviation so that I can choose that model for the prediction or deploying this model or applying this model. So right now what we have done is we have generated the model and we are validating the model. I'll just show you more parameters how to validate this model. Let the support vector machine also model generation will take over. Meantime, do you have any questions? I don't know. You don't have any questions. Is that, are you able to follow me or not? That is something very important I would like to know. I know this is something new, but yeah, we have to start at some point at some day, right? It's good to start today. So anyway, this video is there in YouTube, so you will be able to refer it. And RapidMiner, you can search online and download their software. And it works in Windows, Linux, as well as Mac. So so make sure that you try this today itself. whatever it's accessible for you. Now, what we can do is, I have already told Biswa sir also, we can give you star drop licenses for you to try it out. So, if you want to evaluate or just test your optimizations using star drop, only to the participants we can arrange an evaluation. But again, it's up to your interest. Whoever is interested, you can contact Biswa sir and we can arrange that for you. Otherwise, rapid minor, just go online, search for it, and you will be able to get it. Okay, so now we can see the support vector machine model is also done, and we could see the correlation here. So the correlation, the best one is the random forest, which is having 0.89. So let's do a certain analysis here, right? So when you're looking at the data, this is what we have already seen, and looking at the statistics of the data. So this is for a specific descriptor BO2, another F04. We can see how it is distributed among the data. And we have N072, then ONV1VPCD. Please read the paper to get to know about all these descriptors. And the log BCF is also distributed. Okay. And here we can see what's the minimum, maximum value, average, and standard deviation. So this is the descriptive statistics data that we can take for each of the features as well as the data that is being generated. And what is a weight by correlation? And this is something very, very important. This is plays a vital role in the feature selection. This tells you how far this particular descriptor is correlating to your BCF, bio concentration factor. So this helps us to understand how far it is very well correlated. So X to AV is only having 0.04 correlation, but log P is having around 79.7% of correlation. So this gives you to talk about how far the correlation between the descriptors and the bioconcentration factor. Now this is called intercorrelation matrix, where it tries to tell you there is something in linear regression we tell. Of course, we didn't do linear regression here, but we have linear models generated. So in linear regression, they tell you if two descriptors or two parameters having greater than 90%, even 85% also of correlation, they shouldn't come into the same model, right? That's a beauty there where we have to select. Sometimes we don't look into all these factors. So this intercorrelation among the descriptors matrix helps you to understand this. For example, you can see here, where is it? PCD and PIPC09 is having a correlation of 74%. If both comes into the same model, for the linear regression especially, it will be having some issues like overfitting issues and other things. So these kinds of statistics is something very important. I didn't talk about any machine learning. It's all statistics we are still talking about. So make sure before getting into machine learning, you know statistics in good manner. Now, let's analyze the random forest, because random forest was a model which was giving us a better correlation with a lower standard deviation. So this is the random forest they have generated, how they are being related with the tree nodes and other things, and the weightage based upon the random forest. So here you can see the log P is having around 82% of weightage over the BCP, like BCF, that is bioconcentration factor based upon random forest. Okay, and we can simulate it. I'll come to this a bit later. Looking at the performance, RMSE value, that is root mean squared error value, it's 0.6. Yeah, percentage, squared error is 0.4, and correlation is 0.89. You can see a lot of errors being generated here. and the possible prediction influence. So green color tells you it has more influence on that prediction. The yellow color or orange or red color tells you less significance on the prediction. So that's what it is telling you. For example, in statistics, we say student t test. So student t test tells you what is the significance of that particular parameter into the response variable. Then we can have a prediction chart. This is a scatter plot which tells you the experimental values versus predicted value. So this is called a scatter plot or correlation plot where we used to put in the publications, right? And then we have a production model which can be used for deployment. So I have already deployed this model. So let me show you. So deployed means I wanted to make use of this model of random forest for predicting the properties for my property, predicting the properties for which I am looking at. So this will be giving a lot many data, but rather than going into these data, this I have already shown you all this data. But let me go to a simulator. Simulator means, for example, I have a structure with all these descriptors I have calculated, and I'm saying I have a structure with a log p of this particular value and b02 of this particular value, and on is of around this particular value. What is the bioconcentration factor? It is 2.95. So I did not give any structures here. I did not give anything other than that. I just calculated the descriptors using Padel or any other tool, and I just gave the descriptor values on this particular simulator. It is able to predict the bioconcentration factor. Did I do modeling now here? No. Already I have done it and kept it ready and I deployed it or I applied the model. So this is one way of understanding how I can predict the bioconcentration factor for my molecules which I have not yet synthesized, or for the molecule they have not yet reported anywhere. So I took it from a database. I calculated the descriptors. So this is for each molecule you can see, but you can always load the CSV file directly and predict it also. Now, I wanted to know, I want to get an optimal balance of all these properties. So I'm asking the machine learning algorithm itself to optimize and tell me which could be the best value of these descriptors in order to get the good bioconcentration factor. So I can either mention specific value or I can say maximize it. And then next, I can define the constraint. I can say whether to stay within the two standard deviation and above all attribute minimum and maximum value. So standard deviation also we have to define. And now I can limit the time for, because more the time more optimization. So that's the beauty of optimization parameters, because if it is going to, if you're going to parameterize a lot, it takes time, but taking more time means it has done all the possibilities of permutation and combination and come up with the best solution. If you don't have too much time, then it might come up quickly with something what is possible. So here I'm not saying any limit, I'm just asking it to run. So it is trying to run an optimization algorithm and to see which could be the best descriptor values to get the maximum bioconcentration factor. Literally, it is not maximum. We can go for minimum because this is a log value. So I'll go for the minimum and the next run. So it run the optimization. So in order to get minus 0.23, I need to have the descriptor values of these combinations. So based on these combinations, you can give it in star drop. Of course, star drop capabilities are not there in RapidMine. I told you, no, lead optimization techniques are not available here. So going to star drop, you can say that I want bioconcentration factor this value minus 0.238. Now, please come up with transformation on my existing molecule. So I want to know which could give me something close to minus 0.238. So StarDrop will do the same way what I have did for toxicity to reduce the toxicity to come up with different replacements. StarDrop will be able to take care of that. And it will come up with new replacements or transformation to say whether it is able to get a similar bioconcentration factor. So this is how we do lead optimization. So we did a QSAR, understood the prediction ability. we applied it on our new structure, or we try to optimize and understand what should be the minimum bioconcentration factor within the applicability domain. That's again one more very important point. Okay, so within the 779 structures I selected for that particular model with a lesser standard deviation, I can achieve minus 0.238. So I don't want to go outside the applicability domain. So considering this, now I want to find out what are the novel molecules I could design for lead optimization. And that is completely based upon StarDraw. Now, if you think this is too much advanced for me to take it, sorry, we have to start doing this because already the world is ahead of us in certain cases. I don't generalize that everybody is not doing. Most of you might be doing it, but still, let's everybody do it so that all of them can collaborate each other. And it is not that only a data scientist can do this job. No, it's very simple. As I showed you, you are the best people to interpret which is right, which is wrong, because these descriptors are very well known to you. What is bioconcentration factor known to you? And a chemist knows what could be the structures that could be designed based upon the predictions coming out from star drop. So the combination of informatics, chemist, as well as modeler gives you a good productive novelty in your drug discovery projects. Okay, do you have any questions? I am almost done, but if you have any questions or if you want me to repeat something, I can do that. But Dr. Biswa already mentioned me because you all require some live demo. That's why I took a very interesting case study to give you a combination of optimization as well as applying ML models to optimize the property and bring it to design the structure. So if you have any questions, I'm happy to take it up. We have another 6 more minutes left. I think I'm on time.
Sir, we have received one question, sir. You can view
Yeah.
on your screen.
Sir, I
Can
have
you view,
already
sir?
told answered this. I have already answered
Um,
this question to Mahendra
okay, okay,
that for
okay.
docking we have Autodoc, Vina, Esmina and all, and paid we have CSAR, FlexX and all. And then if you're going for QSR, we have like Paidal software to generate descriptors. We have 9 KNIME, which I showed last time during the last FDP. And then in order to generate the models for QSR, today I showed you Rapid Miner. Rapid Miner is free for academics. You do not have to pay any penny. So generate the descriptors using Paddle, bring it to Rapid Miner to generate machine learning models. And if you want to do a lead optimization, unfortunately, we do not have any open source available. Mostly a command line, so I don't know how comfortable you are. So we have StarDrop, but it's commercial, where you can bring these modeled data optimized parameters to generate. So from property, you can generate new structures, which is synthesizable.
Okay, sir. Thank you. Uh,
Thank you. Dr. Mamata, I don't know whether you are aware of Udemy, Udemy. Udemy is a platform which has a lot of training videos for software, right? And you can go to Coursera. Don't look for certificates. You don't require certificates. You're basically going for learning for your research, applying it for research. So Coursera, you have an option to audit the courses, and Udemy has many free courses where you can learn rapid miner and all these things. Now, if you look at StarDrop, a drug discovery hackathon, there is a YouTube channel for drug discovery hackathon. If you go there and search for Optibrium or Biosolvite, you will get around four different videos for the training on these softwares. And for the 9, you can always go to my YouTube channel where I have spoken about 9 as well as extensively on QSAR using payload descriptors also. So these are all plenty available in YouTube. So I would recommend, rather than searching in search engines, you can go and search in YouTube. Most of these video tutorials are already available there extensively. And the slides today I presented is available in SlideShare. So let me show you that since it is already shared the screen. So slideshare.net/GiriBio. So today's whatever I have presented, including many other presentations, are available in this particular link. So if you go to this particular one, machine learning in chemistry and drug candidate selection, if you go to this slide, you will find the exact slides which I presented today. Okay, and if you want to know about um the demo videos on 9 and QSR, you can go to my YouTube channel where I have given much more extent, because it's each videos are 2.5 hours. Now, if you think, no, I want only 10 minutes video, sorry, I cannot explain 10 minutes a QSA. It's lot many to learn. So many of the tutorials and software trainings are given here, including similar presentations are given in this channel. And today, whatever I have used in RapidMiner and Orange for QSR, that could be found in GitHub, where you can also use GROMACS, protein modeling. All the particular notebooks are also available in the GitHub. So if you go to this page, scroll down, you will be able to see QSAR 9. Then in the aging data, you will find some of the deployed QSAR models to predict anti-melanogenic activity. If you go to MD notebooks, you will be able to find parameters and Python scripts. You need not to write any scripts. It's just to run. You need to just replace the PDB ID. That's all you have to do. So you can run even you can run molecular dynamics on Google Colab. That means you can learn molecular dynamics using your mobile also. You don't require any computer. You don't require any graphics card. Nothing is required. You have to just go there and click the link. It will go to your Google account, Google Colab. It will not copy any data of yours, but you will be able to learn GROMACS using Google Colab in your mobile. So if anyone is interested to learn molecular dynamics without any computer, because many of them complain that I don't know Linux, I don't have computers, but you don't require anything. You will be able to do that and perform it online. And it does including, so I have included all the result analysis, also including graph, all the final graph creation. You can see RMSF, RMSD, all graphs will be created, including visualization is available within this. So GROMACS have already cited this particular notebook, so they have validated it. So at least you can use it for learning purpose. Yes, that's all.
Thank you, sir, for your nice presentation. So you have given details about this uh, artificial intelligence and how to apply this in our research field. It will be very beneficial for the participants, sir. We are very much thankful to you, sir.
Absolutely, sir. Thank you
Thank you,
so
sir.
much. Best wishes to everyone. Thank you.
Yes.
Thank you, Dr. Girinath G. Placer, for sharing your vast knowledge and ideas on 2D and 3D lead optimization using computational tools and showing a live demo of QSR ML modeling softwares. Definitely, it will be very much helpful to all the delegates and participants of this EFDP. May I now request our respected Principal Sir, convener of this EFDP, Professor Dr. BVV Ravikumar Sir, to felicitate our eminent speaker with an e-certificate.
Uh, Thank you, Dr. Girinath, for the valuable information about lead optimization using computational tools, and also for the demo. And first of all, thank you very much for accepting our invitation again, and you're one of the important person who, because of whom our FDPs are running successfully. Thank you very much, sir. And on behalf of Roland Institute of Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Please accept it, sir.
Thank you so much, sir. It's my honor. I'll be happy to accept it. Thank you.
Thank you, sir, once again.
Thank you, sir.
Thank you, sir.
Thank you, sir. Best wishes. Take care. Bye.
Dear delegates and participants, for the next scientific session, which will start at 2 p.m. for today, kindly join us with the link to be provided in your WhatsApp group and email ID. Thank you, one and all.